All right, so we've seen how the information makes its way first from DNA into RNA, and we're going to focus on mRNA again for the protein coding genes. So now we need to figure out how the information in the mRNA gets translated into a different language, into the language of amino acids that will form the protein. So we're going to talk about what we call the genetic code. Now, the cool thing about this genetic code is almost universal. So that means everything from a bacterium to a plant to a fungus to you have the same kind of code of coding information. Now that's really cool, right? And that hints at our shared lineage, but that also means that you can take sequences from any organism and put them into any other organism and they will work. That's really cool, and we will see that is used to make like things glow with the GFP protein, which is one of my favorite proteins. So this universality of the genetic code is really, really cool and really useful for biologists. We're going to talk about the steps in protein synthesis. How do we build a protein? And we have to talk about the role of the ribosome in doing this, because that's what's actually building the protein out of this. So we're talking about translation. Translation is decoding the information in the mRNA and turning it into an amino acid chain. The amino acids get joined together by peptide bonds. So a peptide bond is the bond that forms between one amino acid and the next amino acid. Now, for talking about amino acid chains, which we'll call a protein, technically it's a polypeptide. Once it's all folded up, it becomes the fully mature protein. The peptide bonds here form between the amino acids. So here's one amino acid, here's the next amino acid, here's the next one, here's the next one, here's the next one. We have a way of naming this direction. We call the front end the amino terminus, or the N-terminal end, because it's got this amino group on it. And then we work our way down to the carboxy terminus, which is the C-terminal end. That is the end of this. So instead of using that 5' and 3', which applied to DNA and RNA, here we have the N-terminus, which is the front of the protein, and the C-terminus, which is the back. Okay, there are several different types of RNA, as we've seen. We're going to focus on messenger RNA here because it contains the information that'll get translated into a protein. But we're going to need two other types of RNA to help do that. We're going to need tRNA, which stands for transfer RNA. That's going to bring the amino acids into the ribosome and decode the mRNA by binding in these groups called codons. We'll see that in a sec here. We're also going to use rRNA, which is ribosomal RNA. This is part of the ribosome. It helps catalyze the peptide bond formation. Now, we've talked previously about other non-coding RNAs, microRNAs that regulate gene expression. We talked about SNRPs, the short nuclear riboproteins. Those have snRNA in there. But we're just going to class all other RNAs as other non-coding RNAs. There's a lot of them. We're still discovering them. We don't know what all of them do. So translation. Translation is the process that we're taking the information from the mRNA into the protein. And all protein synthesis, making of proteins, requires this translation process. And it is done by the ribosome. And it forms what we call a peptide, a polypeptide. So here we have bacterial examples. Remember, in bacteria, we don't have a nucleus, so the process of transcription, DNA to RNA, and translation, RNA to protein, can happen at the same time. So here we see a mRNA, and if you imagine enough, you can kind of see the mRNA in there. It's very small, right? This is an electron micrograph, but these molecules are real small. These blobs here, though, these blobs, the dark ones, are ribosomes, and here's a model of the ribosome. The ribosome is building a protein off of this. And you can see as it works its way down the mRNA, these protein chains are longer and longer. Now here there's a whole bunch of ribosomes translating this one single mRNA here. So in bacteria, this process happens at the same time. In eukaryotes, it will be separated by not being in the nucleus. It either happens on the endoplasmic reticulum or out in the cytoplasm. So how does this all work? How does the ribosome take the RNA information and convert it into protein? Because we have a bit of a problem, right? We have a language barrier here. There are 20 amino acids, but there's only 4 DNA bases and 4 RNA bases. So how do we get 20 different amino acids with only 4 bases? This was a very interesting question. And again, this is one of the reasons people thought protein was the genetic material for a long time. But there was a whole series of experiments. Again, I encourage you to take my Micro 250 class. I talk all about them there. They were very elegant experiments. But what they worked out is that 3 groups of letters from the RNA, so these 3 bases together, were enough to code for all of the amino acids. So if you think about it, if you just have the 4 letters, right, you could code for 4 amino acids. If you work in groups of 2, that's 4 times 4, you could get 16 amino acids. That's not quite enough. But if you work in groups of 3, that's 4 times 4 times 4. You get 64 different, what we call codons. Codons are groups of 3 bases together, and the different codons code for the different amino acids. So we have 64 different codons, and you can see here, this is a codon table. I will always give you codon tables. If you need them, you don't have to memorize this. I don't have it memorized. There are a few key ones that are nice to know, but let's look at how we read this. Okay, so you're reading along, and your first base in the group of 3 is, let's say, an A. So we come down here and we go to A. Then we go up here and we go along. Let's say it's another A. So we have AA, and where that intersects, we see the codons here. If our third base was AAA, that would code for the amino acid lysine. If it was AAC, that would code for asparagine. The different codons code for different amino acids, and you will see there's some redundancy here. So you could have CCU for proline or CCA for proline or CCG. There's kind of a reason why. I will show you that in a moment, and we think evolutionarily this is kind of advantageous. You can actually have some mutations happen and it won't actually change the amino acid sequence, which is a good thing, right? Because if your protein's functioning, you don't want it to change. Now, there are 3 very special codons that we need to talk about. The first one is AUG, which codes for the amino acid methionine. It is the only codon that does this, and it will be the codon that is at the start of translation. So that means all proteins will start with methionine. There could be other AUGs in the protein, and there'll be methionines, but all proteins should start with methionine. So that's like our number 1 rule in translation. And you're about to learn how to translate our sequence here. So you're going to be doing this in just a second. Remember, you always start with an AUG. Okay, if you're not starting with an AUG, if you're not starting with a methionine, you're going wrong. Okay, there are also 3 other codons, UGA, UAA, and UAG, which you will see say stop. These will stop the translation process. There is no amino acid that is coded for there. I'll show you what happens there in a moment. But let's talk about this overall kind of structure here. Okay, we're about to see it, but the way that the cell recognizes these codons is through base pairing with a special tRNA. So the tRNA is an RNA that folds on itself, and it has a site called an anti-codon that matches up with the codon sequence. And attached to the other end of the tRNA is the amino acid. Now, we call the genetic code here degenerate. That's because there are more than 1 codon per amino acid. It's partly because of this hypothesis called the wobble hypothesis, where we think that the tRNA, when it comes in and binds, this third position doesn't need to quite match up perfectly to bring the amino acid in. So it could be UCC, so let's do that. UCC, we get serine. Okay, or it could be UCU, it's also serine, right? That allows some flexibility here. So if you were to have a mutation here in the third site of a codon, it wouldn't actually affect what amino acid gets coded for. That will be important. We'll talk about that in the mutation bit in just a moment. All right, so what are these tRNAs? Well, tRNAs are one of our non-coding RNAs, and they fold into, at least in 2 dimensions, this kind of clover leaf shape. They actually fold back in base pair on themselves. That's kind of cool, but it actually forms 2 structures that are really important. Down here at the bottom, this loop, the anti-codon loop, is where it actually matches up with the codon in the mRNA. This is what is actually decoding the information. On the other end is the specific amino acid coded for here. So the tRNA is bringing in the amino acids. That's why it's called transfer. It's transferring those amino acids to the ribosome. So each tRNA recognizes a specific codon, given that wobble as well. And it's got the specific amino acid on the other end. Now, when you actually look at the 3D structure of the tRNA, it looks kind of more L-shaped. And here's the space filling model. It's kind of this R or L-shaped thing. And here's the site where the amino acid sticks on. And here's where the anti-codon is down here. So it'll fit into there. All right, so we have the basics here. Let's go ahead and throw you to the wolves. It is time for you to decode the information of life. This is kind of maybe one of the coolest things you'll ever do, right? You'll be able to read the genetic code now. I think that's pretty powerful. So here is your mRNA. Here is your codon chart. Okay, I want you to remember a couple slides back what I said about there's a couple special codons. Remember those rules. So go ahead and pause the video and try to translate this protein. All right. Hopefully you were able to do translation no problem. Now, I'm going to start here with my translation of this. So what we need to do is remember our first rule here, right? We said that AUG is the start codon. All proteins should start with a start codon. Now you will notice that the mRNA does not start with that. That is okay. There is nothing wrong. All we need to do is act like a ribosome and scan along until we find nope, not quite, AUG. All right, there we go. There's our first start codon. So that's where we're going to start translating. So we want to scan along until we find our AUG. Now this part at the beginning, that's the untranslated region. It doesn't get translated. AUG is going to be where we start. Okay, and remember, AUG codes for methionine, so all proteins should start with methionine. That is indeed the case. This is going to establish what we call the reading frame. Okay, so here is our first codon, AUG. Then our next codon in the reading frame will be AAA. Go ahead and pause the video if you need to, and continue translating this if you perhaps didn't start with the AUG. Okay, so our second codon, right, is AAA, then GCA. It can be helpful to underline every other codon. So AAA and then underline this one. So let's go ahead and work through them. AAA, we come over here to A and then A and A. That is lysine. So our next amino acid would be lysine. And then GCA. Here's G, here's C, and down here is GCA, alanine, so on and so forth. Our next one is CUC, so CUC, that's leucine. And then we come to UAA, so we go UAA. All right, here we go. This is the stop codon. This is where translation stops. So our mature final sequence here is methionine, lysine, alanine, leucine, and that stops. You'll notice there is a 3' UTR, an untranslated region at the end as well. These are very common and exist in reality. Okay, so let's look at the whole thing now, the whole central dogma. So we started from the DNA, right? We have the DNA, which acts as a template to build the mRNA that gets translated into a protein. Now remember back, there was a second strand of the DNA, the coding strand. it didn't actually participate. But you'll notice ATG, AUG, the coding strand contains the message, and we can decode it using a codon table. So when we actually look at DNA on the computer or something like that, we always look at the coding strand because we want to know about the proteins that are in there, assuming we're working with protein coding genes. All right, so this is how we decode, translate the information. But how does the cell actually do it? Well, I gave you a little hint with the tRNAs there, but the real kind of structure that enables this to happen is the ribosome. The ribosome is used to translate the mRNA into amino acids, and it is a mixture of that rRNA and protein. The rRNA forms an important catalytic part of this structure. Ribosomes are made up of two parts. There's a small subunit and a large subunit. And just so you've heard the terms, these get measured in terms of not really size, but kind of weight. This 30S and 50S subunit for prokaryotes, the 30S is the smaller and the 50S is the larger. This S stands for Svedberg units. It has to do with how things get pulled downwards in centrifugation. So these Svedberg units aren't actually additive. So the prokaryotic ribosome is actually only 70S in units. It's not 30 plus 50, which is 80. It's 70. It has to do with how it is. Why we still call them this is historical. I want you to have heard the terminology. You don't have to understand the centrifugation part unless you go on to more advanced biochemistry. But knowing that there is a small subunit and a large subunit is important because they come together and they do different things there. In eukaryotes, we say the ribosome is 80S. Now you'll notice 40 plus 60 is 100. That's not 80, right? That again is the whole Svedberg unit thing. 40S is the small subunit and 60S is the large subunit. So ribosomes in prokaryotes and eukaryotes are slightly different. That's kind of a good thing because if you want to make an antibiotic that kills bacteria, you can actually target their ribosomes and not kill yourself in the process because your ribosomes are slightly different, which is the whole basis of medicine, right? It's trying to destroy the pathogenic microbe and not kill the patient at the same time. So what do the two pieces do? The small subunit here in my cartoon diagram, the bottom piece, the small subunit is what actually binds to the mRNA. The larger subunit here is actually going to bring in and have slots for the tRNAs that are going to do the decoding. So here's our space-filling model of it, and here's our ribbon diagram. You can see how complex this structure is. There's both rRNA and protein in here. All right, so let's learn how the ribosome functions. Okay, the ribosome starts as the small unit, and we're going to look at bacterial ribosomes here. The ribosome scans along the mRNA, the small subunit scans along until it finds the AUG start codon. Okay, then it brings in the first amino acid, which has methionine on it. Okay, side note, if you take microbiology, you will learn about in bacteria, the first methionine has a special additive on it. It's called a formal methionine. We don't need to worry about that. All I want you to know is that it is methionine here, so that's why it says FMet. Okay, so we scan along until we find the first AUG, where methionine gets added. This methionine is attached to a tRNA. So here's the anti-codon, here's the amino acid attached to the tRNA. Okay, when that happens, the large subunit of the ribosome comes in, and you will notice there are 3 sites, and they just so happen to fit tRNAs perfectly. These 3 sites are E, P, and A. I will tell you what they are in the next slide. Okay, let's start in this direction because this is kind of how they move, actually. They move right to left in this diagram. A is the acceptor site. The acceptor site is where new tRNAs can enter, and they actually test if they match. So it's not magic, like a tRNA comes in and it tries to match up. If it doesn't, it gets kicked out until finally it does match, and then it gets added to the growing chain. Now this happens super fast because these are molecules and they're moving at incredible speeds. Okay, so the acceptor site brings in new tRNAs, tries them out, tries to match them up. The P site, that is the peptide site. That is where the amino acids are going to get joined together. the peptide bond, which sticks one amino acid to another amino acid, is going to be built. And then we have the E site, the exit site. This is where the spent tRNA goes and gets ejected from. So let's watch this process. Okay, so we have our methionine here. Then our next tRNA comes in. If it matches, we then form a peptide bond here, which is catalyzed partly by the rRNA that's in there. Then we shift the whole ribosome down. Okay, this is more detailed than we need to know, but there is a GTP-containing compound that comes in and moves the thing over. You got to spend energy here. We shift this over. So this first tRNA scoots over to the E site, and the second one scoots over to the P site, and we have a slot for the new tRNA to come in. Okay, and so this whole thing just kind of ratchets over 3 bases at a time. You'll notice these sites are 3 bases wide. That's how the codons get decoded. Okay, so it's going to keep doing this over and over and over again until it reaches a stop codon. How does it know to stop there? Well, there's no tRNA that is coded for by the stop codons. Instead, what happens is a protein called a release factor comes into that site and causes the protein chain, the polypeptide, to be cleaved off. It gets released. And then the ribosome falls apart, it dissociates, and these pieces can go back and translate a new mRNA. So there's no tRNA for the stop codons. There instead a release factor comes in and causes the complex to fall apart. So that's translational termination. The 3 codons, UGA, UAA, and UAG, don't have a tRNA. We release the protein and let it float off. It's going to go do some things here in a sec. The ribosomal subunits fall off the mRNA and translation is complete. Those subunits can be reused. They'll find another mRNA and go and translate that. Or let's watch an animation of this process.
Protein synthesis occurs in 3 phases: initiation, elongation, and termination. In bacteria, such as E. coli, initiation requires the association of several components: small proteins called initiation factors, the first tRNA, the mRNA to be translated, and the small subunit of the ribosome, also called the 30S subunit. The key event of initiation is the association of the mRNA and 30S subunit so that the start codon, AUG, is centered on the ribosomal subunit. The 50S subunit docks to complete the initiation complex. Initiation uses energy by hydrolysis of GTP. Note that the ribosome has 3 tRNA binding sites: an acceptor, A site, also called aminoacyl site, a peptide, P site, and an exit, E site. The ribosome is ready for elongating a chain of amino acids. Elongation requires an elongation factor called EFTU, associated with GTP, to bind to aminoacyl tRNAs. This complex enters the A site. Correct selection of the tRNA complex depends on codon-anti-codon pairing. Here the anti-codon CCC pairs with a GGG codon. GGG codes for the amino acid glycine, which this tRNA carries. Next, the ribosome catalyzes the formation of a peptide bond between the 2 amino acids, and the hydrolysis of GTP drives peptide bond formation. The resulting EFTU, GDP, is expelled. An elongation factor called EFG, associated with GTP, binds to the ribosome. The GTP is hydrolyzed, providing the energy to ratchet the 50S and 30S subunits ahead 1 codon. This maneuver opens up the A site, and the uncharged tRNA slides into the E site. The next aminoacyl tRNA that enters the A site creates a conformational change in the ribosome that telegraphs through to the E site and ejects the uncharged tRNA. These elongation steps repeat along the mRNA. Eventually, the ribosome arrives at the end of the coding region, marked by one of 3 possible stop codons. This phase of translation is called termination. No corresponding tRNAs exist for stop codons. Instead, a protein called a release factor, which has the general shape of a tRNA, enters the A site. The release factor triggers hydrolysis of the peptide tRNA bond, thus releasing the peptide. The 30S and 50S subunits fall off the mRNA and separate in the cytoplasm, available once again to form a new translation complex with another mRNA.
All right, so that animation has more detail than we need, but you can see the basics of how the ribosome ratchets along and the tRNAs come in, decoding the message. So that's how the cell decodes the message. I think that's really, really cool, right? That allows us to understand how information is stored in DNA, how we can convert it into mRNA, and then how we can make that into a protein. So we've talked about how that code is universal, right? This codon table here is universal. All organisms share the same codon table. That's really powerful, right? That allows us to take DNA sequence from one organism and put it into another. Because this process, this transcription and translation is all universal, that allows us to do some fascinating things. So I want to tell you about mRNA vaccines, which you've probably heard about. They're rather new, but the principle is very simple. So a vaccine is a way of training our immune system to recognize a pathogenic microbe before we ever encounter it. Why do we want to do that? Well, we want to build antibodies against those pathogenic microbes so that when we do come in contact with them, our immune system is ready to pounce on them and we don't get as sick or don't get sick at all. That's kind of a good thing, particularly if, like, say, the microbe is going to kill you, right? So we like to create these vaccines to train our immune system. The immune system recognizes proteins from the microbe and builds antibodies against them. Let's take the case of a virus. Let's say it's SARS-CoV-2, which causes COVID-19. SARS-CoV-2, the virus has these spike proteins. The spike proteins, in and of themselves, do not cause sickness. What the spike proteins do normally for the virus is they bond to our human cells, they bind to the receptors that are on there, and they trick their way into the cell. Okay, so our immune system eventually catches on and makes antibodies against the spike proteins. But unfortunately, immune systems are slow, so it takes a while for them to catch on. That's why you get very sick when you're infected with SARS-CoV-2 and you feel like garbage. What if we could train our immune system to recognize this spike protein, but we don't have to get the whole virus into our cell, which causes cellular damage, your lung tissue primarily. So what if we could train our immune system on this? Well, we could just show our immune system the protein, but actually making proteins in a test tube is kind of difficult. Getting them to fold properly and function correctly is tough. So what we can do is we can utilize the central dogma of molecular biology to help us with that process. So if we take the mRNA sequence that codes for this spike protein, and we package it up in a little lipid nanoparticle, that lipid nanoparticle will fuse with our membrane. Remember, our membranes are made of lipids. That will bring the mRNA into our cell. Now, when your cell sees an mRNA, it goes, that is an mRNA. I'm going to translate that. So that's what it does. It translates the mRNA into protein. And your cells make these little spike proteins, and they put them on the outside of the cell. And your immune system goes, hey, that looks like something from a virus. I'm going to build a defense against that. And that swelling and inflammation you feel when you get the COVID vaccine, that is your immune system going, hey, I'm going to start making a response. You're not actually really sick. You're just responding to it. That's what's happening there. So you build that response up. Then your immune system has all these antibodies. When you encounter the real virus, it's ready to pounce on it and destroy it before it can even make it into your cells. And your cells do not die then, right? When a virus goes into your cells, it kills them. That's why your respiratory tissue is damaged when you have COVID-19. So this mRNA vaccine is really, really flexible. Our previous methods of making vaccines would have been taking this spike protein and adding it to a non-harmful virus that can infect us. There's some pros and cons to that, but the great benefit of this mRNA vaccine technology is that we can change this sequence real fast. In theory, we could change this in an afternoon, okay? Whereas in the past, making new vaccines took months and months, possibly years. We can update vaccines much quicker. That's really important because, as we've seen, SARS-CoV-2, the coronavirus, has been mutating, right? The spike protein mutates and our antibodies no longer respond to it. So every year, we probably need an updated COVID vaccine to respond to the newest spike proteins. That is made much easier by working with the mRNA, because we can work with mRNA no problem. It's super easy to work with. So this mRNA vaccine technology takes advantage of the central dogma and the universality of the genetic code. That's pretty sweet, I think. All right, so once the polypeptide, the chain of amino acids, is made, that's not the end of things, right? We talked about our 4 levels of protein structure. Proteins need to fold, and that folding is partly dictated by the amino acids that are in there, but also the environment that the protein is folding in. Some environments are not favorable for protein folding, so sometimes the cell needs to put the protein in the right environment to get it to fold. There are little structures, they look kind of like a little barrel basically, called chaperones. These chaperone proteins are just that. They're like a little barrel. We stick the polypeptide in there, close it up, and it creates a favorable environment for folding, and the correctly folded protein pops out, okay? And now it's able to go function. We also modify proteins. Oftentimes we add things like a signal sequence to the end of the protein. The signal sequence gets added to the N-terminus, and that kind of directs the protein where it's going to go in the cell. So it might be like you're going to go to the lysosome, you're going to go to the plasma membrane, you're going to go to a mitochondria. It's like a signal that tells the protein where to go. There are other modifications. Things can get added, like methyls or phosphates, or other types of molecules can be added to the proteins. You'll learn more about that in upper level courses. All right, so we're going to finish up with a bit of review. Please pause the video right now and translate this sequence into the protein. So I'm giving you an mRNA translated into a protein. Okay, pause the video. All right, so first step, right? Scanning along. We're looking for an AUG. There we go. That's our first codon, and that establishes our reading frame. So AUG, CCA, AGU, UAA, and I can already tell because I have them memorized that that's a stop codon, okay? The only 4 codons I actually have memorized are AUG and then the stop codons. So you don't need to memorize any of them. I will always give you a codon table. All right, let's do the translation now. So AUG, that's our methionine. That's where we start. CAA is glun, which is glutamine. Next, we have AGU. So AGU, that's serine. And then UAA is our stop codon. All right, perfect. So we now know how to decode the genetic information. So remember, there's 4 different types of RNAs that we've talked about. In this case, we were talking about mRNA, which contains the message, the tRNAs, which brought in the amino acids and decoded the message. The rRNA, which is part of the ribosome, helps decode this. In other contexts, we've talked about other non-coding RNAs. Those are mainly involved in gene regulation. The information is stored in 3 base groups called codons. Each codon specifies a different amino acid. The cell recognizes codons because tRNAs have an anti-codon that pairs up with that codon, and the tRNA brings in the correct amino acid with it. So to translate a sequence, scan the mRNA until you find the first start codon, AUG, and then start working along in groups of 3. Remember, all proteins will start with methionine. Now, I'm telling you this as a biology teacher. There are occasions when proteins don't start with this. It's extremely rare, though. So for our purposes, they always start with methionine. Continue on in those 3 base paired codons until you reach a stop codon. The cell uses the ribosome to do this process. It's made up of rRNA and protein. It's got those two subunits and then the different sites that accept the tRNAs, and it kind of walks along the mRNA translating it. After translation, proteins need to get folded and they often get modified, sometimes with that signal sequence. Okay, so that's how the message is decoded. In the next section, we're going to talk about what happens when mutations occur and that can affect the message that's coded for.